Assalamu alaikum. Thank you for being here today. I'm Zirwa Rahat and I'm here to talk to you about next generation sequencing. History of next generation sequencing. First sequencing technique were developed in 1977 by Frederick Sanger and Walter Gilbert. Sanger sequencing based on the chain termination method that was adopted as a primary technique in the first generation. At that time, DNA sequencing was a labor consuming and required radioactive material. Years later, in 1987, Applied Biosystem introduced the first automatic sequencing machine, which made the process much faster and accurate. This project and X Prize competition stimulated the development of the next generation or second generation sequencing. In 2006, era of next generation sequencing, 454 sequencing, Illumina, etc. While the second generation of sequencing became widely popular in basic research, new improvements in sequencing technologies, and open an era of the third generation sequencers, introduction of next generation sequencing. It refers to the deep, high throughput in parallel DNA sequencing technologies. The next generation sequencing technologies are different from the Sanger method. Millions to billions of DNA nucleotides can be sequenced in parallel, yielding substantially more throughput and minimizing the need for the fragment cloning method that were used with Sanger. In that, they provide massively analysis, extremely high throughput from multiple samples. The parallelization of a high number of sequencing reactions by next generation reaction sequence was achieved by mini as a restoration of sequencing reaction. Types of next generation sequencing. There are basically two types of next generation sequencing, a sequence by synthesis and sequencing by hybridization or ligation. Pyrosequence. It is a method of high throughput DNA sequencing that utilizes a single strand of DNA with a length of 4 to 500 bp. Ion torrent sequencing, a technology that directly translates chemically encoded nucleotide bases into digital information that translates chemically encoded nucleotide bases into digital information on a semiconductor chip. This approach marries simple chemistry to semiconductor technology. This technology gives results more simpler, faster, and cost-effective and scalable than any other technology. Scolex, a technique that is used to determine the series of base pairs in DNA. It is also known as DNA sequencing. The reversible terminated chemistry that concept that was invented by Bruno Canard and Simon Serfert at the Pasteur Institute in Paris. This method based on reversible di-terminators that enable the identification of single nucleotides. It can also be used for whole genome and region sequencing, small RNA discovery. Helicos. It was the first commercial next-generation sequencing implementation to use the principle of single molecule fluorescent sequencing, a method of identifying the exact sequence of a piece of a DNA. PacBio. It captures a sequence information during the replication process of the target DNA molecule. Then we have a look on basic characteristics of next generation sequence. The principle behind next generation sequencing is similar to that Sanger sequencing, which relies on capillary electrophoresis to process millions of reactions in parallel, resulting in very high speed and throughout at a reduced cost. The next generation sequencing method uses array-based sequencing, which combines the techniques developed in Sanger sequencing. The genome sequencing projects that took many years with Sanger methods can now completed in odds with next generation sequencing. The steps of next generation sequencing. It involves three steps: library preparations, sequencing, and data analysis. Library preparation. A first step in next generation sequencing, it allows the DNA or RNA to adhere to the sequencing flow. It allows the sample to be identified. There are basically two methods of library preparation: ligation-based and tegmentation-based. Sequencing. During the sequencing step of the next generation sequence workflow, libraries are loaded onto a flow cell and placed on the sequencer. On most Illumina sequencing instruments, clustering occurs automatically. In a process called sequencing by synthesis, the clusters of DNA fragments are amplified in a process called cluster generation that results in millions of copies of single-stranded DNA. In a process called sequencing biosynthesis, chemically modified nucleotides bind to the DNA template strand through natural complementary. Each nucleotide contains a fluorosecond tag and a reversible terminator. The fluorosecond signal indicates which nucleotide has been added. After the reading the forward DNA strand, the reads are washed away, and this process also repeats for the reverse strand. This method is called paired end sequencing data analysis. After sequencing, the instrument software identifies nucleotides, a process called base calling, and the predicted accuracy of those base calls. During data analysis, you can import your sequencing data into a standard analysis tool or set up your own pipeline. Today, you can use initiative data analysis apps to analyze next generation sequencing data without bioinformatics training or additional lab staff. These tools provide sequence alignment, variant calling, data visualization, or interpretation. Technologies overview of next generation sequencing. Helicos Bioscience, Life, APG Solid System, Pacific Biosciences, and Terran Technology. Advantages of next generation sequencing. It reduced sample size, low reagent cost, picking, transformation, vivo cloning, high degree of parallelism, then capillary sequencing. Applications of next generation sequencing. It enables metagenomics, discovering non-clonic RNA, gene regulation analysis, exploring chromatin packing, mutation discovery, transcriptome analysis, molecular diagnostic for analogy and inherited disease study. Thank you so much for your listening, for your attention.